Welcome to our second podcast in Chapter 4. We're going to be continuing with the third week, moving into the fourth week with the process of neurulation. We're also going to talk about the formation of somites and the beginning of the formation of the intraembryonic body cavity. On the right, again, you can see that we've got our bilaminar embryonic disc, but then we also start to move through processes where portions, these medial portions of our ectodermal cells get very tall compared to the lateral portions, and they begin to rise up along what's going to be another groove. This is going to be our neural groove. They rise up from a median point. There is another bend point, which we call the dorsolateral hinge point, that's going to cause this to bend back over onto itself on both sides, and that's going to create our dorsal hollow nerve cord, or our neural tube, and that's going to be the process of neurulation. So here you see a couple of snapshots of that neurulation process. We can see the notochord underlying, and remember that notochord is one of the prominent inducers of this process of neurulation and the inducer of the ectoderm becoming our neural ectoderm. Let's watch a short video of the process of neurulation, and we will return here to discuss the details. We start with a review of primitive streak and gastrulation. Here you can see the primitive streak forming, and remember this is in the posterior marginal zone. And as our embryonic disc increases, when you look in the anterior region along the midline, you will see another ridge of tissue. This is going to be our neural groove, which is forming, and the light blue is going to be the raised sides that are going to be our neural folds. These are going to rise up, they will fold over, and they will contact each other and fuse along the midline, and you can see that medial fusion happening. And the last points of closure are going to be at the cranial and the caudal ends. Those remaining openings are called the neuropores. And so that's going to basically zip itself closed. We have passed our third week, moving into the fourth week, and by the end of the fourth week of development, we have completed neurulation. We have folded the embryo, which we'll talk about in the next unit, and we have something recognizable now. It's not just a flat sheet, but a three-dimensional embryo. As we saw in the video, neurulation is the formation of our dorsal hollow nerve cord, or the neural tube, and this is going to give rise to the brain on the anterior cranial aspect of the embryo and the spinal cord on the more caudal or the posterior aspect. So we had the neurula, the solid ball phase, then we went into the hollow ball or blastula phase. Once gastrulation began, then we were the gastrula, and now quickly following that, we are now in the neurula stage, when we can refer to the embryo as the neurula. Now, as we begin to look at a lot of these processes, understand that some things are happening at the same time. As gastrulation is finishing, neurulation is beginning. So, especially when we get to our organ development, everything's happening about the same time. And most of our organ systems begin development about the fourth week. So by the end of this time, our other organ systems are starting to develop as well. So we're just going to have to take them one step at a time, one piece at a time, and understand there are a lot of things that are overlapping. So when we left looking at gastrulation, we had our ectoderm on the surface. We had talked about the inductive power of the notochord to influence these medial overlying ectodermal cells to become neural ectoderm, or neural plate ectoderm. These cells are going to get much, much taller than the lateral ectoderm, and so their location is they're going to be above the notochord, or dorsal to the notochord, and they're going to be more cranial than the primitive node, right along the midline of the embryo. Now, from our ectoderm, we can see that we are certainly going to give rise to our surface ectodermal structures: hair, nails, subcutaneous glands. Our neural tube is what we're focusing on now. This is going to be our central nervous system, motor neurons, retina, spinal cord. Then once we form this secondary population of migratory mesenchymal cells, some people almost refer to this as a fourth germ layer. These cells then are going to give rise to the remainder of our nervous system, our adrenal medulla, melanocytes, A lot of the structural features in our head and neck are also going to come from these neural crest cells, and we'll talk about how they form relative to the developing nervous system. But as we move along, we look at this neural tube, and it's basically going to be formed by two really driving factors. And we're going to have this right and left half that are going to come together, as we saw in the video, to form our neural tube as a whole. But we also are going to have differences that occur in the cranial or the posterior aspect of the body versus the posterior. So we've got the cranial anterior, the posterior caudal, and in the primary neurulation, this is going to happen in the cranial part to about the position of our hind limbs. Now, when we talk about a quadruped, we can talk about forelimbs, hind limbs. Humans, we have arms and legs, but in embryo, we distinguish forelimbs, which will become our arms, and hind limbs, which will become our legs. Now, secondary neurulation is going to occur from about the sacral vertebrae to the end. So there's not a lot that's going to happen with the posterior, but they're really two separate foci of neurulation that's happening in adults. So primary neurulation, we're going to focus on this. The secondary, we're not really going to spend much time on. But with primary neurulation, first we form our neural plate. Again, this is going to be induced by the notochord. Cells are going to get much taller. Typically, we might have fairly flat to cube-shaped epithelial cells. But for our neural plate or neuroectoderm, these are going to be very tall. They're going to be columnar. And when we look at our existing embryonic disc, especially this cranial portion, as much as 50% of the ectoderm is going to become neural plate. Now, it's not that 50% of your body is central nervous system, but at this point in development, half of that ectoderm is going to be involved in making neural plate. The lateral half is going to grow to populate the rest of what we will create with our surface ectodermal structures. Now, the neural plate, we have to form it, then we're going to shape it. And what's going to happen is the plate cells are going to, as they migrate, as they populate, as they undergo mitosis, they are going to lengthen. And they're going to lengthen preferentially in an anterior-posterior direction, not laterally. Now, we can have a process where we have convergent extension. And so what's going to happen is these cells are going to intercalate. They're going to interdigitate themselves together, overlap, in other words. And as they overlap, they're going to make the plate longer with this preferential anterior-posterior axis elongation, yet they're going to become more narrow. And that's going to assist in pulling these cells toward the middle, and it's going to assist in creating this depression that we're going to see as the neural groove. Now, once we form our neural plate, these cells get taller on either side. We're going to have the bending of the neural plate, and this is going to form our neural groove. This is going to be a lot like the primitive streak, just on a much larger scale. So along the very median plane of our neural groove, we are going to have an anchorage point, and these cells are going to be called median hinge point cells. They're anchored to the notochord, so they're not going to go anywhere. They're not going to get any taller. And so as the lateral cells get taller and taller and taller, this median hinge point is locked in place. And as these cells grow, they're going to create neural folds, getting larger and larger and larger, going straight up, maybe at about a 45 to a 50 or 60 degree angle. Now, after we form these tall neural folds, then along the lateral boundaries, we're going to have other sites. These are dorsolateral hinge sites. Now, these anchorage points are going to be in our lateral surface ectoderm, so they're not going to be able to get any taller or move in any direction. And so the cells that are more dorsal to these hinge points are going to begin to grow, and they are going to fall and grow back towards one another like a wave. The wave goes up, but when you get to the top or the crest of the wave, it crashes over towards the middle. And that's what both of these crests or waves of neural ectoderm are doing. They're crashing back towards each other and toward the neural groove. So let's see how this, we don't have an animation for this. I wasn't able to find one. But here we can see our neural plate forming, cells getting taller. The middle, we're still not as tall as we are just on opposite sides. But there you see the median hinge point just above the notochord, and you see how that's keeping the middle part of our neural plate from getting any taller. But as the cells preferentially grow, then on the lateral aspects, they are getting taller and taller. Here we have our neural folds on either side of the crest, and you can see that in the scanning electron micrographic view that we saw earlier. Well, as we get taller and taller, we finally get to this point where we have our dorsolateral hinge points. You see those kind of in the olive drab color. And in the scanning electron micrograph, you can easily see that hinge point. There's almost a 90-degree angle at this point in the scanning electron micrograph for some of these, but certainly about a 45, 60 degrees, something in that range. But do you see how it's bringing the top of our neural folds crashing toward one another? And they've begun to color that top part a different color, and we're going to see why in just a minute. But as the crests continue to close together, they're going to contact one another, they're going to fuse. And that topmost part that was in green, that is referred to as the neural crest. And you can see that the surface ectoderm in blue has contacted each other, sealed itself off, and the green neural crest have distinguished themselves and separated themselves into this new population, and that is our neural crest cells that we saw in that previous image, or this honorary fourth germ layer. And then at the very end, once that's all closed off, we have our surface ectoderm completely separate from our isolated neural tube, and our neural crest cells begin to migrate away from that site throughout the body. Now, this closure point that happens, we've got the hinge points bringing everything together. Well, the fusion, in our video, it looks like it happens in the middle and zips up and down. And for some species, that is in fact the case. But in humans, there appear to be 3 to 5 different sites where the neural ectoderm is going to contact each other, and it's going to start zipping anterior and posteriorly from all of those points until we get to the final 2 places that still have to be closed, and that's going to be the rostral, which refers to your nose, or the cranial neuropore, and the caudal neuropore. We saw those openings in the video where the head and then when the tail would be. Those are the last places that zip up for the closure of the neural tube. Now, as we bring this tissue together, as these crests start coming together, what we're going to see is that neural plate cells have cell adhesion molecules, and this is what keeps them tightly adhered to each other. And one of these cell adhesion molecules is referred to as E-cadherin, with the E standing for epithelium. So our surface ectoderm here has these epithelial type cell surface receptors, and that's what holds them on one to another. But then as we see the crest starting to fold over, we see that the neural tube cells begin to express N-cadherin. Now, N for the N-cadherin stands for a neural cadherin. And then we're also going to see N-cam, a neural cell adhesion molecule. So as these cells are crashing over the top, the epithelial cells that are going to create the closed surface, they continue to express E-cadherin. But the cells of the neural tube begin to express N-cadherin and M-cams, which are not compatible with each other. So the ectodermal cells adhere to one another, the neural cells adhere to one another, and since they have different cadherins and cell adhesion molecules, they don't adhere to each other. That separates the ectoderm from the neural tube, and that's what helps us close it off, seal it off, and have it completely separate. Here's a scanning electron. I believe this is a frog embryo, and you can see neurulation happening. We see our zip point here in the middle, and then we have our openings, which would be cranial to the right, caudal to the left, and those are going to be our last 2 points. And the last small openings are going to be our neuropores. This is just sort of a synopsis of that, and I apologize for the haziness. I couldn't find this picture again. But you've got the plate, then you have the shaping, and then you have the closure of the neural tube and the surface ectoderm, resulting in completion of neurulation, and that will be by the end of the 4th week. Now, I want to show this image as well because as we talked about the regression of the primitive streak. You can also view that here as we look at neurulation. So as we form our neural folds, you can see how extensive the neuroectoderm is. Remember, possibly 50% of our ectoderm. But now you can see how less significant the primitive streak is just by 21 days. The embryo is outgrowing that posterior tissue that is not really going to contribute any structures to the developing individual. It's going to be everything that's going to be cranial to that primitive node that's going to create the individual. So I just wanted to throw that in as well for that perspective of the regressing of the primitive streak. So neurulation completed by 3rd week. Closure of the neural tube is going to be completed by the end of the 4th week. Now, we saw in a couple of the illustrations our neural crest cells. Some refer to them as neuroectodermal cells. And these cells, as they form at the very crest of that wave, they are going to lose their adhesion for each other. The N-cadherin neural tube cells, they stay attached. The E-cadherin cells, they stay attached, form the ectoderm. The neural crest cells, however, are going to express different cell surface receptors that allow them to be migratory. And they are going to be able to migrate dorsolaterally. Now, they're in a dorsal position. And so I kind of hate to use that term because as you see the population forming here, this is dorsal. But as they lose their adhesion, they're going to be migrating laterally. But many of these are starting to move in a ventral direction away from this dorsal area. So here we can see our distinguishing cell types. These are our definitive neural tube cells. The lighter color is our surface ectoderm, and then our neural crest cells are beginning to distinguish themselves at the molecular level so that by the time we get to the closure point, we have the ectoderm coming together, the neural tube coming together, and the neural crest cells form and then migrate away from that zone. Now, neural crest cells, when we look at the molecular events that happen, we see that neural plate, neuroectoderm, they can be specified as neural crest. If specification, remember, is you take them out, put them in a tissue culture dish, they can become what they normally will become, in this case specified as neural crest. And what we see is it's a couple of factors. We got bone morphogenetic protein again, these BMPs. They're going to be expressed by neural groove cells, but we have another very potent transcription factor and a very potent morphogen, Wnt6. This is going to be expressed by our ectoderm. Now, these are very much controlling through development. We've seen many of these already. Wnt6 is a new one. But what we're going to see is that these two factors, the BMPs and Wnt6, from the ectoderm and from our neural tube, they are going to induce the neural crest cells, and they're going to express what are referred to as neural plate border specifiers. Now, these are transcription factors, PAX3 and Distillus5. A lot of these came from work with Drosophila. So what we're going to see is that PAX3 and Distillus5, they are going to block neural plate and block ectodermal specification, which basically means we are going to be permissive for neural crest formation. We're not inducing them, we're just preventing the others from forming. A little bit later, we're going to see that specific neural crest specification transcription factors, the TF, they're going to be turned on. So FOXD3, that specifies neural crest. SLUG is a transcription factor that enables the molecular machinery to be turned on that allows for the loss of cell adhesion and the subsequent migration of the neural crest cells. So again, very early on, we have molecular events before we can see the actual histological events happening, mediated by and large by growth factors, morphogens, and transcription factors. Morphogen is any agent that's going to cause a change in cell behavior leading to a change in cell shape or cell form or cell position. So once again, just a walkthrough. Think of this almost like a flip book, showing our ectoderm, our shaping with our hinge points. We can see the different transcription factors here. We got the BMPs in neural tube, Wnt6 in the ectoderm. But there you see FOXD3 and SLUG already being expressed in that third image in our neural crest cells. They're specifying neural crest so that by the time we get to the fusion point, we get to the closure point, they are prepared to migrate away from that neural crest location. Now, neural crests are not the same in all locations. It would be nice if it was, but they weren't. We're going to see that cranial neural crests are really going to contribute, by and large, to craniofacial development and the structural features. So the cartilage bone neurons, glial cells, connective tissue of the face, thymic cells, odontoblasts, these are the tooth precursors, and then bones of the middle ear and the jaw. Those are going to come from our neuroectoderm and the neural crest, specifically from the cranial, some people call them the cephalic neural crest. Now, the bulk of what's left, the trunk, these cells are going to migrate, again, ventrolaterally is what I prefer to call them. Many are going to form dorsal root ganglia, sympathetic ganglia. They're going to contribute to the adrenal medulla, the chromaffin cells there. Those migrating laterally that stay near the dorsal surface or close to the ectoderm, they're going to form melanocytes as part of our epidermis. Now, our cervical or sacral, those are going to be parasympathetic ganglia. And then we have the specialized population that's going to occur near the cranial, near the trunk. These are our cardiac neural crest. Now, some of these are going to migrate into the developing blood vessels of the developing heart. We're going to see that early on in heart development, we have one path getting out of the two-chambered heart. Neural crest cells help partition that one outflow tract into the aorta and the pulmonary trunk. Without the correct migration of these cardiac neural crest cells, they are not going to form, and you're going to have a tremendous problem, tremendous congenital defects that are not compatible with life. So here we have an idea of our neural crest cells. We've got, you can see the location of our cardiac crest, our trunk crest, we got our sacral neural crest cells, and then some of our vagal neural crest cells. That's going to contribute to the different structures that we see. And this is just a peel away. They've peeled back the ectodermal sheet, showing the green arrows of the neural crest cells that are going to migrate to contribute to melanocytes, and then this more ventrolateral migration that we're going to see contributing to our ganglia, our sensory ganglia, and our sympathetic ganglia, as well as the other structures such as our cardiac neural crest. Now, that leads us to yet another major thing happening in this early third week or so of development. We talked about mesoderm formation. We talked about the distinction between our chordomesoderm and our lateral mesoderm. Well, as we look more at this lateral mesoderm, if we can divide the embryo into a cross section, a transverse cut, we're going to see that we have mesoderm that's close to the neural tube, and then we have mesoderm that's further away on the lateral aspect, and we have mesoderm in the middle. So the mesoderm that is immediately adjacent to the neural tube, we refer to that as the paraaxial mesoderm, the axis being the neural tube. And this paraaxial mesoderm is going to begin to distinguish itself and differentiate itself and condense itself into paired blocks of tissue on either side of the neural tube. And these paired blocks of mesoderm are going to be referred to as somites. Now that leaves our other mesoderm. As we move out laterally, the paraaxial gives rise to the somites. Then we have the lateral most mesoderm, and this is going to form a lot of our musculature for the heart, blood vessels. We're also going to form some blood cells from this tissue as well. And then the mesoderm that's in the middle, the intermediate mesoderm, it's not a very creative name for it, but that's going to contribute to our urogenital system, portions of our adrenal gland. And when we look at this in graphical form, they highlight very well the paraaxial mesoderm. Here we have our developing, this is our notochord, neural tube. This is our paraaxial mesoderm. Our lateral mesoderm is out here, with the intermediate being somewhere in the middle, and it's difficult in this image to really see that. We'll see that as we develop and we produce our somites. We'll be able to see that a lot better. Here we have a breakdown of our different mesodermal derivatives. We've got the chordomesoderm you see in red, developed into the notochord. Our intermediate mesoderm that goes to our urogenital system. Paraaxial mesoderm, that being primarily our somites. We're going to contribute to head structures. We've got our cartilage, tendons, muscle, skin, endothelial cells, and we're going to break these somites down into these different regions as we go through them. But then our lateral plate mesoderm, we're going to contribute musculature to the body. We're also going to contribute to our circulatory system, and these terms splanchnic versus somatic. Somatic refers to our body wall. Splanchnic refers to our viscera. Much like parietal and visceral as far as peritoneum, somatic body, splanchnic refers to our viscera. So we'll be using those terms as well as we go through this material. So somite formation, near the end of the third week, we are going to begin to see somites produced. And again, like much else, the molecular mechanisms driving this, even before we see them histologically, we're going to see that we begin to express BMPs in our unsegmented paraaxial mesoderm, and that the BMPs actually get downregulated by a transcription factor that's called noggin. As noggin downregulates BMP, that leads then to the formation of our somites. Now, these blocks of tissue, somite is just the name for the whole thing. But we're going to see that we have a sclerotome portion of the somite, which gives rise to the vertebrae and the ribs. Myotome, myo of course meaning muscle, the musculature of the back. Dermatome, dermis of the back. We now have a new one. This is called the syndotome, and this is going to give rise to connective tissue that are the tendons. And we're going to see that most of our axial skeleton and musculature are going to come from our somites. So here we have a scanning electron micrographic view. They have removed the surface ectoderm. That's what you see here. We're looking at the underlying mesoderm. Here we have our neural tube. Here we have our paraaxial mesoderm that's unpaired. Now we're beginning to see the creation of these paired block-like structures that we know as somites. Now, the formation of somites is going to lead in many instances to the staging of developing organism. I know for chick embryos, the number of somites will dictate what stage of development, how many hours it has been since fertilization. And really, it's going to be like clockwork. And so in chicks, when you look at a chick embryo, a pair of somites form about every 90 minutes. And you're going to have about 50 pairs formed for the totality of chicken development. And this wave-like structure, not only is it BMP being downregulated by noggin expression, but we're also going to see fibroblast growth factor, FGF, is going to play a role because it's going to set that boundary of the somite that is going to cause it to have a rostral and caudal movement, so that somites are going to form first toward the head, and they're going to form later as you move down the tail. Kind of throughout development, that's not an uncommon theme to see structures form first in the cranial or anterior part, and the structures form later as we get toward the posterior part. Now, as we're forming somite, again, all of this is happening at just about the same time, so we sort of have to take these as we go. And so the paraaxial mesoderm, this is where we're forming the somites. But as we look at our lateral mesoderm, or lateral plate mesoderm, we can see that we're beginning to form some fluid-filled spaces. Apoptosis is going on. We're producing water and causing cells to move apart. And these little islands, these coelomic spaces, are going to continue to grow and get larger and larger and coalesce together until we basically form our intraembryonic coelom. And in this case, we pull back the ectoderm. At this point, we're looking at the mesoderm underneath. What we're forming is this horseshoe-shaped cavity. And this is going to be our intraembryonic coelom, that through the folding of the embryo, this is going to give rise to our pericardial cavity, our pleural cavity, and our abdominal cavity. But for right now, it is simply a horseshoe-shaped body cavity. Now, what we see as we divide that mesoderm in the lateral plate, because of this horseshoe-shaped intraembryonic coelom, We're going to divide it into the somatic and the splanchnic mesoderm. The somatic is going to be continuous with the amnion and attached. The splanchnic is going to be adjacent to the yolk sac. So again, as I've mentioned before, you can think of these as parietal with our body and visceral with our internal organs. Splanchnic in this case associated with a yolk sac. This is going to be associated with the endoderm and our developing gastrointestinal tract. So in this transverse section here we have our intraembryonic coelom. On top, adjacent to the amnion, we have the somatic, and beneath we have the splanchnic associated with our umbilical vesicle or our yolk sac. Now, some terminology that I want to pass on as well. The somatic mesoderm, if we consider that somatic mesoderm and our ectoderm on the surface, it's often referred to as the somatopleur versus the splanchnic mesoderm plus the endoderm underlying that as the splanchnopleur. Again, this has to do with our somatic versus our splanchnic mesoderm and the underlying germ layer. Now, we've been talking all this time about this little embryonic disc that is occurring right here. We haven't said much about what was going on with the trophoblast at all, or here in the chorion. But you can see we've got a nice connecting stalk that's connecting the amnion and the umbilical vesicle back to our trophoblast. And remember, this is cytotrophoblast and syncytiotrophoblast. This makes up our chorion. But what we're going to be developing now, our cytotrophoblast, they have been busy. They have been dividing tremendously, and they have been protruding as these solid cords into our syncytiotrophoblast. Here you can see a lot of the lacuna within the syncytiotrophoblast where we have maternal blood flow, the uterine gland secretions. And so these chorionic villi, these accumulations of cells, are protruding into the syncytiotrophoblast. And if we were to cut one of these in cross-section and look in the very middle, what we're going to see is we're going to see a population of cells that are mesenchymal, these pluripotential mesodermal cells. And as long as that's all we see, then we are going to call this a secondary chorionic villi. The primary chorionic villi are solid cores of cytotrophoblastic cells. But when those cells give way for this mesenchymal core, we now refer to them as secondary chorionic villi. Now, a little bit further in development, you can see we have got a tremendous explosion of our cytotrophoblastic cells. They're actually going to create a shell that's going to completely surround this developmental unit. They're going to completely surround the lacuna that are forming in the cytotrophoblast. So we're going to create a cytotrophoblastic shell. Now, within our villi, those mesenchymal cells are going to undergo a process that is called vasculogenesis. This is the formation of blood vessels from stem cell or precursor tissue. Angiogenesis is the sprouting of new blood vessels off of existing vessels. But we're going to form blood vessels from our mesenchymal tissue. And as soon as you see capillaries within this chorionic villi, now we refer to that as a tertiary chorionic villi. Solid cytotrophoblastic cells are primary, mesenchymal core is secondary, but blood vessels inside are our tertiary chorionic villi. Now, I think you can see by this image what we are creating. We're beginning to see the blood vessels coming from the embryo out into this extra embryonic mesoderm and coursing through our chorionic villi, and they're coming very close to those maternal lacuna. We're really starting to create our uteroplacental flow so that now our embryo can be nourished by the nutrients in the blood from mom instead of simply by diffusion through its own circulatory system. So at this point, what we have is an arteriocapillary network in its beginnings of our extra embryonic circulation. So here we're looking at our cytotrophoblastic shell. It's going to completely surround our chorionic sac. And these cells are also going to form a much firmer attachment to the endometrium. This is going to hang on very tightly, and that again is going to be the foundation of our placenta. Now, the last thing to mention as we look at these villi, you're going to have some villi that are going straight down into our cytotrophoblastic shell and into the endometrium. Well, these are anchorage type of villi. They're going to hold on very tightly, increase that surface area with those cell adhesion receptors. These are referred to as stem or anchoring villi. But we have others that are going laterally, and you can see these coming very closely opposed to our lacunar network. These are going to be our branch villi. These are the ones that are the exchange of materials between fetal and maternal blood. So as we look at our chorionic villi, again, primary, secondary, tertiary, here we only have cytotrophoblastic cells in the core. We add mesenchymal cells, we become secondary. We add capillaries, we're tertiary. And as long as we have capillaries, if we're anchoring into the endometrium, we're stem. But if we're lateral and adjacent to our maternal blood in the lacunas, then we also can be considered branch villi. And that concludes our Chapter 4 podcast. We'll see you in Chapter 5.